for people who couldn't attend, and I think I am. Okay, great. So today's lecture might not be fully covered, so Brittany will notify me when I am like 10 minutes before the end, because I do want to let you go earlier. That's just my policy. I believe that students deserve to, you know, chill out a little bit. So I'm going to finish. If I don't cover everything today, I will cover it next, like on Thursday. And you probably saw my announcement. I modified, like I uploaded new lecture on e-class. I just reduced the content so it can be easier for you to follow. Okay. No, this doesn't. Okay. Anyway, so can you all see? Yes, you got it. It's an error that I need to press. Okay. So what are we going to talk about? Today we will give a brief introduction about evolution in general. And this will be literally the only class where I cover evolution totally. I'm more focused on diversity, but in terms of evolution. But there are some basic concepts that you have to know. So we will briefly mention possible conditions on the earlier, like on the last, like how I told on earlier. In the process, we'll talk about the process that gave the rise to first cellular life, which you will see later has very nice name. And we will mention how two main branches are bacteria and archaea diverse, like the pathway for diversity. And we will end up with the process of endosymbiosis, which is actually the basis for arisal of third lineage, which are we. So the origin of Earth, I'm sure you cover in other classes, so that's why I will just briefly go through it. And what I really want you to know, and I will try not to mention too many numbers because I hate them, but I do want you to know that Earth was formed 4.5 billion years ago. So that's when our planet came up. And the way planet came up, again, you'll learn probably in other classes if that's your major. I'm not going to go in details, but I will briefly mention that the old star exploded, supernova form, and supernova like formed its energy formed from explosion of the star. It released dust and gases, and that release of gases and dust created this cloud. And in that cloud, our sun was formed. When stars started to compact, that caused release of light, of heat and light, and then there was exposure to gravitation and collisions, and that started to compact and fuse, and that's how we actually got planets, and planet Earth is one of them. And this is just so. Again, I will ask in first quiz Hadean, Archean, Proterozoic, and Phanerozoic. But I will exactly tell you what you want to know. So obviously you need to know that Earth was formed 4.5 billion years ago. And I do want you to know that number. That number is you will figure out why it's important. So during the process of planet forming, energy was released and heated the emerging Earth. And the reason why is that important to mention is that Earth was very hot in the beginning. It wasn't very suitable for any organism. So it was literally hot magma. As Earth was cooling down, that led to formation of cracks and rocks. And to be quite honest, there are studies that show that Earth was bad place to live like 500 million years. So it was formed, but there was no life. Water at the time was present only as a vapor, so you can imagine that life could not be. But who was the first one on Earth? How we know that microbial life was actually present on Earth? So we know based on these fossilized cells in rock, it's quite hard for microbes. It's not as easy, well, I don't want to say easy, but it's easier to conclude about evolution when you can actually get dinofossil. With microbes, we rely only on carbon and sulfur in rocks, which are indicators of early microbial life. And those rocks that are 3.5 billion years old and younger contain microbial formations that are called stromatolites. So it is important for this class, and this is something that I will obviously ask, that what are stromatolites? So those are fossilized microbial cells that we find in rocks, and they are evidences of early microbial life on Earth. This is literally all what I will ask you, at least at this class. So again, stromatolites, fossilized microbial mats, what do they contain? They contain filamentous prokaryotes that are trapped in the sediment. Where can we find them? Marine basins, hot springs. Those old, ancient stromatolites are formed by phototrophic bacteria. And there is a good evolutionary-based reason why are phototrophic bacteria, simply because phototrophic bacteria were living on Earth when we did not have oxygen. Modern organic stromatolites are formed by cyanobacteria. And again, there is a reason for that. The reason is that once when oxygen evolved, which we will cover in next few slides, bacteria and organisms that like oxygen could develop. So it was basically diversity based on the available molecules on early Earth. Now, very often we get the question, so who was the first? Where was the origin of cellular life? So, okay, Earth was hot, no, nothing could live there, but it was cooling down and then life was formed. So who was the first? How was the first cell formed? And more important, how that cell looked like, what that was. And this is what we will intensively cover, and I will repeat that through like this is something that is very important. I want you to finish this class with knowing how first cell arise and what was the first cell on Earth and how it looked like. In those terms, we will cover three basic substrates, and the last but not least, RNA world and protein synthesis. And the third one, we believe, is considered to be correct. Now, what is the surface origin hypothesis? So this hypothesis, personally, to me, really didn't make any sense, but there were people who are more experts than I who talked about this. But basically, people would say that there was something called primordial soup, and that soup contained organic and inorganic compounds in the warm bond. Okay, easy to imagine. And then the first membrane enclosed self-replicating organism just originated from that soup. Okay, what was the evidence? Well, there were evidences about spontaneous life formation if you have organic and inorganic molecules. Yes, that all makes sense. I could imagine there is a pot full of organic and inorganic compounds. They interact together, they form membrane, and they stellarize. Where is the issue with that? The issue is that fact that Earth was actually quite bad place to live. It was really hot. So it wasn't really something that can host any organism. So that is the only reason why I personally do not believe that this hypothesis is accepted. But we do not need to know what was the basis for it. Subsurface origin hypothesis actually makes more sense. And what does that mean? That means that life originates from below the surface, like beneath the ocean floor. So, well, in that instance, there are many hydrothermal vents. So, but then beneath of that ocean surface, like those that hot hydrothermal vents were happening, even though it was very hot, it was stable. So conditions did not fluctuate a lot. And that's the main reason to believe why it was maybe possible to work like this. Because again, organisms evolved, adapted to the hot environment. unlike surface hypothesis where we just don't have any more evidence that it actually works. Now, in these sites beneath the ocean surface, there were many inorganic and inorganic compounds, and then naturally here hydrogen and hydrogen sulfide. Again, I'm using the word reduced inorganic compounds. This might be a little confusing for you because if you didn't take 311 with me, then you have a problem. You cannot connect immediately electron donor and electron acceptor. But you will figure out later as we are going through. The point of these two compounds that were available beneath the surface of the ocean is that they are full of energy. They are source of electrons. So whoever lives there can take out those electrons and start transporting that electron, and that transport forms energy. Obviously, you have to have electron acceptor, but you will get that later as we are moving forward. The point is that beneath that ocean surface, first of all, conditions were stable. Second of all, there was supply of resources. Like there were energy sources there. So it makes total sense. That's what, so think about now that there is a warm water over there, like hot, and that water actually flows through the crust of the surface and mixes with cold ocean water. So here is where chemistry comes into the game. So beneath the surface, you will have hot. Hot water will be alkaline. And then when that goes through the surface, like crushes it, it reacts with very cold acidic, now acidic, so you have basis acid, ocean water. And that ocean water had a lot of iron. And that iron interacted with molecules that were from beneath the surface and formed these structures like pyrites, silicates, carbonates, and magnesium-containing formations. Now, these all minerals in pores that contain iron and nickel, those are mostly sulfides. I will ask you to know about pyrites, because that's iron-containing molecule, iron sulfide. And now this iron compound, and let's only stay with iron, but there are other elements as well. This iron now behaves like takes the role of catalyzer, like enzymes are now. And they catalyze, but this is not enzyme. So they catalyze formations of amino acids, simple peptides, sugars, bases. So you can see that we actually now have a basis for formation of cell as we know now. Phosphate, which was present in seawater, helped in formation of RNA, but nucleotides actually polymerized first into RNA. So this leads us, so that subsurface hypothesis leads us to the currently accepted hypothesis of the RNA world. So again, iron formations, catalyzed formations of amino acids, nucleotides, all building block polysaccharides, all building blocks of primary molecules. And that's how we got RNA. So how did this self-replicating system arise? So we call this RNA world. So what RNA world means literally is that first self-replicating system were RNA molecules. So not DNA that is currently accepted as the stable molecule, RNA. Why we think it's RNA? Well, RNA is fragile. It's very possible that it survived in gel-like formations at the ocean floor. And it is a very small molecule, but it has catalytic activity, which is great. And we have some evidences now that RNA molecules have catalytic activity. So we think that it's possible that RNA catalyzed its own formation from available molecules there. Also, RNA binds amino acids. So again, another evidence. So, and again, we know about ribozymes, which are RNA molecules that can catalyze formation of simple proteins. So the moment when you have proteins, you have the most important part. Cell can keep developing. Different proteins were made on early Earth, and Now, when RNA helped in forming these different proteins, now these proteins actually can encapsulate spirits and other metal-containing formations. So you see what we have now. We have semi-permeable membrane. So something that coated something else, something that surrounds something else, encapsulates. Now, as more and more proteins were formed, they now took over catalytic roles. So now we have actually enzymes that are doing that have catalytic role, unlike RNA, which is a little bit pushed back, but has other significant role. Why RNA did not involve as a primary molecule for genetic information storage? Because DNA was more stable. It has double strand. It's double-stranded. RNA is unstable. So that DNA RNA protein system was fixed in early cellular evolution, and it's the basis for processing of biological information. Now, we briefly mentioned this cellular membrane. So when RNA catalyzed formation of simple proteins, those proteins just coated pirids and other metal formations. But we do know nowadays that our cell membrane is mostly made of lipids. Well, actually, it's mostly made of proteins, but it has lipids at all. So next important step definitely was to design those lipids, like to build that phospholipid bilayer. So phospholipid bilayer precursor, so precursor of phospholipid bilayer are membrane vesicles. Those are, and we see many organisms nowadays forming vesicles, which are literally teeny tiny structures that are just separated from the membrane. So cells use that even nowadays to export something from the cell, like a cargo delivery. So those proteins that were embedded in a lipid membrane, they actually form that semi-permeable structure. In earlier time, when the first cell was formed, those vesicles were made to push out nutrients in and waste in and out of cell. Obviously, nutrients in, waste out. Now, but the big picture from this, what I want you to memorize is that first self-replicating organism might be formed after lipoprotein structure encapsulated DNA and RNA, because this is what cell is. Membrane that has lipids, proteins, and surrounds nucleic acids. How we call this early structure, this first self-replicating organism? Last universal common ancestor, or LUCA, which is literally my son's name, just with a K. And I will never forget it, definitely. So after the cellular life evolved in different directions, so after that, cell life evolved in different directions, different biochemistry based on available energy resources in different environments. But mostly we see, so LUCA diverged to two branches, which are bacteria and archaea. And again, different types of bacteria and archaea have different modes of evolution based on the environment where they are living. So they created different adaptations. How do we know that bacteria and archaea are first two branches from LUCA? What is the indicator? Well, the indicator are phospholipids in membranes, simple as it is. Why? If you look bacteria and archaea, again, two branches from LUCA, bacteria have in cell membranes glycerol-3 phosphate, basically. Bacteria have glycerol-1. So this is different stereochemistry in two organisms, which tells us that enzymes in both branches evolved differently. They just took different pathways, and they were synthesized from different and they synthesized different molecules in bacteria and archaea. Again, bacteria 3P, archaea 1P, and that's how we know that those two branches actually evolved, that those two branches developed from LUCA. Now, What about early metabolism? What was happening with these bacteria and archaea that evolved? Well, first of all, you have to know that there was no oxygen at the time and the Earth was hot. So only specific type of organisms can live there. Oxygen actually showed up much later in something called great. So it showed up with cyanobacteria, but actually became the most important during great oxidation event, oxygenation event. So it appeared, so oxygen was not present until cyanobacteria actually evolved. So there was completely no oxygen. I say no oxygen, but I can also use the word anaerobic because that means no oxygen. And this the breakdown of main milestones in evolution will tell you how the oxygen formatted and when eukaryotes evolved. Because eukaryotes showed up once when there was oxygen. Before eukaryotes, there were cyanobacteria who had to make enough oxygen, right? So these are some numbers that you might have to know, especially the one that Earth formed in Hadean, and how you will know, HAPP, Hadean, Archean, Proterozoic, Phanerozoic. That's how I memorize HAPP, and goes from the bottom to the top. So Earth was formed 4.5 billion years ago, but then there was nothing, right? And then 4 billion years ago, you have LUCA, first cellular life. Great. Then LUCA divided into the bacteria and archaea, and that happened and basically that division gave the rise to some phototrophic bacteria that are called purple and green bacteria 3.5 billion years ago. So phototrophs were right after LUCA. Why? And one important thing, phototrophs, organisms that live off light, they use energy from light. Those organisms, green and purple bacteria, they do not need oxygen. They're great without it. But cyanobacteria are actually making oxygen. They need light. They fix carbon dioxide, but they need they make oxygen. So cyanobacteria were basis for the world that we know today with oxygen. So oxygen was forming, like cyanobacteria were forming oxygen, right? But then that was happening like 1 billion years, actually 500 million years. And then when enough oxygen was formed, you had that great oxidation event. Proterozoic is the era when first eukaryotic organism has been formed, and it was 2 billion years ago. So it was after oxygen was there. What was more important, and we'll see next class, important for eukaryotic evolution is ozone shield, because ozone shield absorbs UV radiation. So eukaryotes could not develop without ozone shield. And in order to have ozone shield, you have to have oxygen. And in order to have oxygen, you have to have cyanobacteria. So it's all logical. Phanerozoic is the last era. And all what I want you to know is that multicellular eukaryotes are made in Phanerozoic, and that's all. That's literally what I'm not going to ask about animals. I'm more interested in microbes. No? In Hadean, which was the first one from 4.5 to 4 billion years ago, like in early Earth, dominant organisms were anoxygenic, which means no oxygen. Those were phototrophic bacteria, green and purple. And dominant form of metabolism was something called methanogenesis. And you will understand what methanogenesis is when we talk about metabolic diversity. So what methanogenesis means is organism that makes methane, gas. As it lives, as it consumes energy, it produces methane. So that was not possible in the early Archean, first half of Archean. But in second half of Archean, actually, oxygenic photosynthesis appeared, which means cyanobacteria. And that was the basis for great oxidation event, which was 2.5 billion years ago. Again, as I mentioned, ozone shield was very important to form eukaryotic organisms, and you couldn't form it without any oxygen. Important is to understand that all those early organisms used carbon dioxide, they did not use free oxygen. And when someone fixes carbon dioxide, we say it's autotrophic, with plants. So you probably heard these terms. These first organisms were evolved to use abiotic compounds as energy source, but as a carbon source, they use carbon dioxide. So mostly we say that first molecules that were used by primitive organisms to generate energy were hydrogen and carbon dioxide. Why hydrogen? Because it's a very great energy source, and it can be used to extract electrons, and electrons are energy. So basically, the types of life that were arising in this period have to have a lot of hydrogen, have to have nitrogen, have to have phosphorus and minerals, and carbon dioxide. That's why we say that they were unoxygenated, no oxygen, they were autotrophs, which means they use carbon dioxide as a carbon source. So is there any organism that functions like this, utilizing carbon dioxide and hydrogen? Yes. And also these organisms, so these organisms that can actually fix carbon dioxide and use hydrogen as an energy source can respire elemental sulfur. Guess what? Elemental sulfur was present in early Earth. So we are quite sure that this was the first metabolism. Unoxygenic, autotrophic, hydrogen was energy source. Usually we say that sulfur was terminal electron acceptor, which you will learn later what it is, but they needed hydrogen and sulfur to develop energy, and they need carbon dioxide to acquire carbon. Where will hydrogen come from? Reaction between iron minerals and hydrogen sulfide. That means that hydrogen will be byproduct. Again, hydrogen is a powerful electron donor, which I'm not going to bother now in details. You will figure out later that hydrogen is actually extremely great fuel that can be used to extract energy to run proton multiforce. Proton multiforce can turn on ATP synthase. So it's an enzyme that builds ATP, and that's energy. Now, basically, the point is that diversity of microorganisms is nothing but consequence of current condition of Earth biosphere. So what does that mean? That means that whatever was available for them to respire and to extract energy from, they would evolve to use it. And that changed chemistry and physiology of all planets. And so basically, it's like a connection between each other. So whatever was available on Earth, they evolved to use to produce energy. Microbes are very unique in terms of taking advantage of whatever is available as energy, carbon, and terminal electron. They will learn how to live in the most extreme environments, which again, we can't do. So we like to say that microbes are chemists of the world because they interacted with available chemistry, but they also changed the chemistry of the Earth because whatever they respire and oxidize, they will produce something else that will change chemical environment. So now when they respire something and produce something else, that something else is molecule that will be used by another microbe so they can live together and coexist on planet. Another thing that I thought it's very important to mention is how the oxygen showed up. And so we know that cyanobacteria made oxygen. And we know that, but cyanobacteria showed up around 2.7 billion years ago. So it was in Archaea. Now, still, great oxidation event was 2.5 billion years ago. So why there is that difference between? So basically, we see that the accumulation of oxygen took like 300 million years ago. So why is that if cyanobacteria already made it? So why it took so much to build oxygen and ozone? So it's very simple. It's very simple. Sorry. Uh, uh, I have to try to figure out how to turn off my notifications on Discord. So, cyanobacterial metabolism basically builds oxygen, but oxygen will react with iron and sulfur compounds. So, in this reaction, iron that is 2 plus will be oxidized to 3 plus, and that iron, 3 plus, will accumulate as banded iron formation. So, basically, that will take oxygen away, so you won't have it anymore. So reaction with iron will take oxygen. So when all abundant iron 2 plus was exhausted, iron was gone, everything was 3 plus, that created banded iron formations, oxygen started accumulating. So it's very simple. And again, another type of what again I already mentioned ozone field. Ozone field was important, but it was important to absorb radiation. So when bacteria and archaea were adapted to some bacteria and some archaea were adapted to anoxic areas, so didn't like oxygen. But oxygen was actually oxidizing so many reduced compounds, and that the term reduced compounds you might not know, but it will be clearly described in the next lectures. But just know that as oxygen was showing up, like cyanobacteria were forming it, now when there were no more iron 2 plus, that availability of oxygen actually limited life and life niche for anoxic organisms. So all those phototrophic green and purple bacteria and archaea were now kind of pushed to the bottom. They could live only where there is no oxygen, and when great oxidation events happened, oxygen was everywhere. So only bacteria that can use oxygen were were able to live in the earth. Again, the rise of oxygen led to the rapid evolution of eukaryotes, multicellular eukaryotes, and animals. And ozone formation was built. Again, it needed oxygen because ozone can absorb radiation of the sun. So again, until ozone showed up, the evolution on the top of the surface could not happen because it was so before ozone formation, everything was happening. It was there to absorb UV radiation, and evolution could happen from the surface. Another important topic that I want to cover is that something that you probably heard about. It's called endosymbiotic origin of eukaryotes. So now we have, so we had phototrophs, unoxygenic. We had all that primitive metabolisms. They were pushed back because cyanobacteria made oxygen. All oxygen started accumulating after iron was exhausted, that we know because we have banded iron formation. Now ozone formed, and there is UV protection, so now eukaryotes can arise. So we know that from Luca we had bacteria and archaea, right? So they went through metabolic diversification many years, but eukaryotes are appearing only 2 billion years ago. And their origin is actually related to the rise of atmospheric oxygen, and we call that endosymbiotic theory. We will cover it in a second. Questions that you usually ask there is, how did these eukaryotes develop, and how oxygen affected their evolution? And this is all explained through endosymbiosis. Again, mitochondria and chloroplasts, which are organelles of modern eukaryotes. Chloroplasts in plants, mitochondria in us and in plants. So those organelles that are now present in us, they originate from stable incorporation of bacteria or archaea into another type of cell, bacteria or archaea. So basically, the host could be either bacteria or archaea, and then ingested another cell, and you got eukaryotes. So that's called endosymbiosis. Oops, sorry. So how do we know that endosymbiosis actually happened? How do we know that some host, bacteria and archaea, actually ate another cell and developed something that we call today organs? Well, there are these four evidences which I will always ask. That will likely be your question in final exam. That's very important. All four of them. These are evidences for endosymbiotic theory. And both mitochondria and chloroplasts of modern eukaryotes contain ribosomes. These ribosomes are different than those ribosomes that exist in cytoplasm. Ribosomes in mitochondria and chloroplasts are similar to, not similar, they are same like bacterial. They have 16S rRNA and 70S sedimentation constant. So mitochondria and chloroplasts are bacteria. The next evidence that we have is that same antibiotics that affect ribosomal function in bacteria also affect ribosomes in mitochondria and chloroplasts. Of course, they're the same, that their mitochondria and chloroplasts are bacteria. Third one is that both mitochondria and chloroplasts actually contain DNA, their own DNA that is completely different than the one DNA that you see in nucleus of eukaryotes. So it's circular, like bacterial. And the last but not least is that there are many other signs of bacterial presence. There are many similar molecules in eukaryotic organisms that we see, and we know that they are connected. One question that we actually didn't respond is, who was the host? What was the nature of that cell? So it could be bacterial and archaeal, can't be anything else. But what was the nature? And second question is, how was nuclear membrane formed? Because remember, eukaryotes have nucleus, and that nucleus is bound by membrane. And for that one, to answer these questions, we have two hypotheses. I personally prefer hypothesis 2, but I will cover both. So hypothesis 1 is given here step by step. So basically, you will have early cell that was developed, and the DNA material will be accumulated there. And this hypothesis says that the nucleus spontaneously formed as DNA was accumulating, and first eukaryotic cell was actually nucleus-containing lineage. And later on, that nucleus-containing lineage ate up mitochondria and chloroplast via endosymbiosis, and we have eukaryotic cell. So that's hypothesis that I quite disagree with that. Second one is hydrogen hypothesis, and this one I think is mostly accepted, but we need to know both. And this one says that early bacteria that was oxygen producing and oxygen consuming and hydrogen producing is the one that gave a rise to mitochondria. And if you think about that, mitochondria are places where that famous electron transport chain and energy production is happening. But host, so the one who ate mitochondria, this hydrogen-producing, oxygen-consuming cell, was archaea. And that archaea actually needed needed hydrogen for energy production. So it was symbiosis. Archaea ate bacterial cell that was consuming oxygen and producing hydrogen. And the reason why it ate it was because it needed their product. It needed hydrogen. So basically, that hydrogen was the reason for archaea to eat that mitochondria. And we nowadays say, nowadays we say that eukaryotic cell is chimera, like made of both bacteria and archaea. And eukaryotes actually have a type of lipids that are found in bacteria, but also have transcription and translation more like archaea. So it's like a combination of both. Energy production, ATP generation, is mostly similar to bacteria. But again, based on transcription and translation, we are actually more similar to archaea than to bacteria. And again, this formation of eukaryotic cell was a really big step in evolution, and that's how we got that chimeric eukaryotic cell, which was very complex. Now, the table that I really like to give you as a question on exams is this table. I want you to understand, in addition to 4 evidences of endosymbiosis theory, I want you to understand similarities between, I want you to understand what eukaryotes got from bacteria, what from archaea. That's quite important. Now, for membrane lipids, remember, bacteria have glycerol 3-phosphate, archaea have 1-phosphate. In bacteria, we call these lipids ester lipids. So ester is the main bond you see in bacterial lipids. Unlike archaea, which will be heterolinked, different. So eukaryotes are similar to bacteria. They are also heterolinked. Who has chlorophyll-based photosynthesis? Bacteria and eukaryotes. You have cyanobacteria, you have plants. Archaea do not have photosynthesis. Histone proteins are not present in bacteria, but surprisingly, you have them in archaea and eukaryotes. Now, that's that transcription translation apparatus that I'm talking about. Cell wall, bacteria have muramic acid, they build peptidoglycan, but archaea don't. Surprisingly, I hope, well, not surprisingly for me, muramic acid is absent in eukaryotes as well. First, tRNA, initiator tRNA is formal methionine in bacteria. Again, it's only methionine in archaea and eukaryotes. Again, anything related to transcription and translation is bacteria and archaea and eukaryotes. So eukaryotes are similar to archaea. RNA polymerases, again, eukaryotes are here unique. They have 3, while bacteria and archaea have only 1. And in terms of sensitivity to antibiotics, only bacteria are sensitive to antibiotics, but not archaea or eukaryotes. And again, Brittany already talked about this, but so because we copy paste slides, so this will be the last slide for this one. You are good to go. And as I promised, I actually finished 10 minutes earlier. Yes, Athena. Yep.
Archaea hosts that have was the first host of the bacteria that had.
It is, for me, it is obvious because it was the early organism, early metabolism. But this was, people had debates. A big problem in microbial evolution, wait, let me just stop. Big problem in microbial evolution is we actually don't have any fossil